So within each of our cells, we have chromosomes. We have 23 pairs, so 46 total, and we get half of these from mom and half of these from dad. Chromosomes are really condensed DNA, and this DNA contains the instructions that our body needs to make proteins. So if we take a little bit closer look at the DNA, we'll see something like this. It is a double helix structure, so it's tightly wound up. And if we take an even closer look, we see that this DNA is made of something called base pairs. So you can see that G's bind with C's and A's bind with T's. So these are called the nucleic base pairs, or shortened as A, C, G, and T. And these stand for adenine, cytosine, guanine, and thymine. So if we take a look at our DNA, we see that we have one strand, and this is going to encode for a protein product. So the way that we turn DNA into protein is that our bodies read it in 3 base pairs. So 3 base pairs encodes for one of the 26 amino acids, and it's specific, the sequence to the amino acid. So our body is going to read this and place an amino acid that it encodes for based on the 3 base pairs. And this chain of amino acids forms a protein. So this is what the sodium ion channel 2 that's encoded by the SCN2A gene should look like for the sake of our video. So now let's talk about what changes can occur that might make this a little bit different. The first we will talk about is called a missense change. This is where one base pair has been changed from what it should be. And as you can see here, the G in the middle has been changed to a C in the lower part. And this changes the blue amino acid to a green amino acid. And this can be written out as the C dot 8 G to C, since the G at the 8th position has been changed from a G to a C. We can also write this as a P dot, so describing the protein change. This would be written as P dot blue 3 green, since a blue at the 3rd position was changed to a green. The next is called a deletion. So this deletion occurs when a base pair gets removed from the sequence. And as you can see, a G has been deleted at the 8th position. And you can see the impact that it has on our protein product. It looks very different than what it should be. If we look at the C dot, we can write this as C dot 8 del G, because a G has been deleted at the 8th position. The P dot for this would be written as P dot blue 3 purple, since a blue was changed to purple. We would also add in FS, standing for frame shift, because a deletion of a base pair means that the 3 base pair frames are going to be shifted. And the last we'll talk about is called a duplication. And so we can see that an extra C has been added on the bottom, and that creates a different protein product. This adds in a green amino acid and also changes the order in which these amino acids occur. This can be written as C dot 7 dupe C, because a C was duplicated at the 7th position. The P dot for this would be written as P dot blue 3 green frame shift, because a blue was changed to a green at the 3rd position, and it led to a frame shift since there was an additional 1 base pair, which shifted the frames in which the base pairs are read. So as you can see, all of these changes have a difference on the protein product that is made. So what exactly can happen when in the protein when there is a change in the gene? So we can have maybe too little sodium flowing through due to a change in an amino acid. We can also have too much sodium being let through due to a deletion of an amino acid, or we can have no movement of the sodium at all. In regards to function, when a sodium channel is overactive, we call this a gain of function change. I want to clarify that when I talk about functionality, I am talking specifically about the gene product or the protein, and not about the person who has this change. So when we say gain of function change, that means that the sodium channel is overactive. Opposite, we can have a loss of function variant that makes the sodium channel underactive or absent. This means maybe too little sodium is being let through, or there is no movement at all. I hope you guys enjoyed this video, and I hope that these illustrations provided you some context. In the next video, I will be going through a genetic test report and showing you where to find important information about the specific change found in you or your child.